Hello everyone, my name is Ankit Gupta and today we will be discussing about the mechanism of urine formation. And I'll start with a brief introduction about kidneys. So here is the diagram of a kidney. Kidneys are bean-shaped, red-colored and retroperitoneally placed organs. The main function of the kidney is to filter out the blood and excrete the unwanted things or other excretory products out of the body. The kidneys are divided into two parts. The outer part, this region is called as renal cortex, and the inner region, inner part of the kidneys, which we can see over here as triangle striated regions. This is the renal medulla. So the nephrons, these are situated in the kidneys. These are the functional unit of the kidneys, and urine is formed by nephrons. Around 13 million nephrons are there in each kidney, and the urine formed by the nephrons is drained out in renal pelvis and which is further moved out of the kidney through ureters. This is the structure of a nephron. The nephrons, it consists of a Bowman's capsule. This is a Bowman's capsule, and this is the glomerulus. These are the fine network of capillaries through which the blood is entering the glomerulus, and the filtration takes place through the filtration bed. So both of them, the glomerulus and the Bowman's capsule, they together are known as renal corpuscle or a Malpighian corpuscle. And rest of the part of the kidney, or sorry, nephron, it is called as renal tubule. The renal tubule, they have different sections in it. The the section which is starting just after the Malpighian corpuscle, this is PCT, convoluted tubule. Then the loop of Henle is there, the descending limb of loop of Henle, and this is ascending limb of loop of Henle. Then comes distal convoluted tubule and collecting duct. So collecting duct of different nephrons, they drain the urine into a common papillary duct, and that papillary duct further drains out the urine into the renal pelvis, as we saw in the previous image. Two types of nephrons are there. The So two different type of nephrons are there. First one is cortical nephron. In cortical nephron, what happens is this loop of Henle, either it is very short or it can happen that the loop of Henle is completely absent. But in case of juxtamedullary nephron, the loop of Henle is there and it is longer, always longer, in order to concentrate the urine. So these are the two different type of nephrons. So the next part, that is the actual topic which we will be discussing, is the mechanism of urine formation. It has three different steps. First one is glomerular filtration, second is tubular reabsorption, and third is tubular secretion. So we will first start with the glomerular filtration. In order to understand glomerular filtration in a better way, we need to look at the diagram of this Bowman's capsule, or uh, diagram of this renal corpuscle or Malpighian corpuscle. So over here, this is the Bowman's capsule. This is the afferent arteriole, which is bringing blood to the capillaries of the glomerulus or glomerulus itself. This is efferent arteriole through which the blood is leaving or escaping the glomerulus. These are the specialized cell you can see over here. These are the podocytes. They wrap around the capillaries of the glomerulus, and both of them together creates a filtration bed through which the filtration takes place. So the fluid that enters the capsular space, this is the capsular space over here. The fluid that enters the capsular space, it is called as glomerular filtrate. And the fraction of blood plasma in the afferent arteriole of the kidneys that becomes glomerular filtrate is filtration fraction. So this is a detailed diagram of a Malpighian corpuscle. Now, this is the filtration bed, which is formed which is formed by these capillaries of the glomerulus. The filtration membrane or filtration bed, it has three different parts. The first one is the capillary itself, the capillary of the glomerulus. It has small pores on it. The pores, these are called as fenestrations. The size of the pore it can be maximum up to zero point 0.1 microns. So this size limitation allows the filtration or the movement of uh, small solutes and the plasma flow, but it prevents the flow of RBCs. So the next part, this over here, you can see this is basal lamina. This basal lamina is made up of acellular material and it prevents the flow of bigger proteins. And the third part is podocyte. This is a podocyte. These are specialized cells. These have feet-like appendages. This you can see over here. These are feet-like structures. These are called as pedicels. And they wraps around the glomerular capillary. This is how they are wrapping around it. They are hugging the glomerular capillary. And the gap which is there between two pedicels, this is called a filtration slit. This over here we can see, this is a filtration slit. So this allows the movement of solutes and plasma for filtrate generation. In together, these three forms a filtration membrane. So this filtration membrane, it creates a leaky barrier. It is a barrier, but it is a leaky barrier through which the filtration process is occurring. And ultimately, the filtration is occurring, or the filtration is driven by different kinds of pressure. So different type of pressures are there which are at play due to which the filtration is occurring. Over here, we will be discussing about the pressures that drive the glomerular filtration. This is an afferent arteriole. This is an efferent arteriole. In by looking at the image, the first thing we can observe is the diameter of the afferent arteriole is greater and the diameter of efferent arteriole is smaller. This difference in the diameter, this reduction in the diameter of efferent arteriole, this creates a resistance in the blood flow, which is coming out of the glomerulus. This resistance in the blood flow, it creates a pressure in it. And this pressure is glomerular blood hydrostatic pressure. So this is a major important pressure due to which the filtration is occurring. This can be up to 55 to 60 mmHg. Next type of pressure is blood colloidal osmotic pressure. This pressure is exerted by the proteins which is present in the blood, like for example, fibrinogen, globulin, albumin. These proteins and exerts an opposing pressure, opposite to the glomerular blood hydrostatic pressure. So this prevents the flow, the prevents the filtration of the filtration process. So this is an opposing pressure. So this is taken in as negative over here. Another type of pressure is capsular hydrostatic pressure. This is the pressure exerted by the fluid which is already occupying the Bowman's capsule. This is the Bowman's capsule, the fluid which is already present in it. This is so that fluid exerts a negative pressure. Again, this is a negative pressure and prevents the filtration process. So altogether, these three pressures, when we will see over here, the net filtration pressure is the actual pressure due to which the filtration is occurring. So ultimately, the net filtration pressure is 10 mmHg. We have to subtract because these two are opposing pressures, colloidal hydrostatic pressure and blood uh, capsular hydrostatic pressure and blood colloidal osmotic pressure. These two are negative pressures and GBHP, that is glomerular blood hydrostatic pressure, is the pressure driving the filtration. So we have to subtract these two from this one. Ultimately, the net filtration pressure which we get is 10 mmHg. And principle of the filtration is the use of pressure to force fluids and solutes through the membrane. So when the fluid gets accumulated in the Bowman's capsule, as we know, it is called as glomerular filtering. As it enters the renal tubule part, This is the renal tubule portion. As it enters, it is called as tubular filtrate. The first section of the renal tubule in which it enters is proximal convoluted tubule. So you can see over here, these are the cells of proximal convoluted tubule, PCT. These are cuboidal, oh sorry, these are cuboidal. They have villi on its surface. These are the villi. The villi are there to increase the surface area, and it helps in maximum reabsorption. So over here, this is the first process of the nephron, that is tubular reabsorption. We will be discussing it in detail over here. So the first thing is reabsorption. What is reabsorption? Reabsorption, it is the reabsorption of all the water, most of the water and most of the fluid which is there in the filtrate, getting back into the bloodstream. That is, in layman's language, we can say that reabsorption is absorbing or going back of the water and lot of solutes which is there in the filtrate back to the bloodstream. So this reabsorption process, it is done by the epithelial cells which are lining the renal tubule. This is a exam. This is over here. This is the Malpighian corpuscle. Only this part, and rest of this is renal tubule. Different sections are there, renal tubule, and the reabsorption process, it is done by the epithelial cell which is lining the renal tubule. So the first section, that is PCT, we'll be discussing about it, and maximum reabsorption takes place in PCT only. So these are the cells, epithelial cells of the PCT. This is the apical membrane of the cell, and this part is the basolateral membrane. This is the renal tubule in which the fluid, the filtrate is there. So these are the villi. These are the so this surface is called as apical membrane. This is the basolateral side, which is facing towards the interstitial space, which has interstitial fluid in it, and the blood capillaries are also there. And two different cells, they are joined together by a tight junction. This is a tight junction, and this tight junction doesn't create a completely perfect seal. It is leaky in nature. So when some solutes are there in the fluid lumen, it crosses the tight junction, reaches the interstitial fluid, and later on, ultimately, they are absorbed by the blood in the capillaries. This type of reabsorption is called as paracellular reabsorption. But in another case, when substances like over here, which we'll be discussing about sodium and glucose, when they moves or when they enter the cell through the apical membrane, they enter the cell and they further goes out through the basolateral side of the cell into the interstitial fluid, and later on they are absorbed by the blood in the capillaries. This type of reabsorption is called as transcellular reabsorption. So in order to understand this in detail, we have an example of sodium and glucose transportation, sodium and glucose reabsorption process. This is facilitated by a transport protein. This is a transport protein over here. This is a symporter protein. Different kinds of proteins are there, transport proteins are there. So three major examples are symporter, antiporter, and uniporter. Symporter are the proteins which are transporting two different molecules in the same site. An antiporter is a protein in which one molecule is getting transported to one side and other is getting transported to opposite side. And uniporter is always in one direction, only one molecule. So whenever a molecule or a substance, it has to be transported through a membrane. That kind of transportation, it can be active or it can be passive. If it is an active transport, it could be primary active or it could be secondary active. In case of primary active, the energy which is driving the transport, it is only and only by ATP hydrolysis. But in case of secondary active transport, the energy which is allowing the transportation process, it is because of the electrochemical gradient of the ion. Over here, this is an example of a secondary active transport in which one of the molecule is getting transported from higher concentration to lower concentration, and another molecule is getting transported from lower concentration to higher concentration. So, why it is like this? As we know that the sodium potassium pumps are there in each and every cell of the body. But in case of the renal tubule, the sodium potassium pumps are there only on the basolateral surface. So this is working continuously, pumping out sodium from the cell into the interstitial fluid. So the inner concentration of sodium is always and always low inside the cell. So what happens is the sodium ions are in higher concentration in the fluid which is there in the tubule lumen, and glucose is in higher concentration inside the cell. So what happens is this symporter protein transport two ions of sodium along with one molecule of glucose into the cell. That is sodium from higher concentration to lower concentration and glucose from lower concentration to higher concentration. So this is a secondary active transport which is taking place. Later on, this sodium is transported out by sodium potassium pump and glucose they diffuses out through a channel, and ultimately both of them are reabsorbed into the blood which is there in the capillary. So in PCT, what happens is glucose and amino acids, they are 100% reabsorbed. 75% of water is reabsorbed. Sodium is 70% reabsorbed and potassium is 75% reabsorbed. 50% of the filtered chloride and 80 to 90% of the filtered SO3 ions and 50% of the filtered urea and variable amount of different ions like calcium and magnesium are reabsorbed. So other ions are also there. We have seen this example as an active transport. Other ions are also there which are passively reabsorbed. Those are chloride, calcium, magnesium, potassium, urea, and water. The water is passively reabsorbed. Whenever the absorption of solute is taking place, the water becomes obliged to follow the solute because water always moves from higher concentration to lower concentration, and wherever maximum solute concentration is there, the concentration of water is lower in that side. So in case when in case of PCT, when the solute gets reabsorbed, water follows it. This type of reabsorption of water is called as obligatory water reabsorption. So this obligatory water reabsorption is seen only in case of PCT and in descending limb of loop of Henle. This is the part. This part is descending limb of loop of Henle. So from here till here, the water reabsorption which is taking place, it is called as obligatory water reabsorption. So next part is tubular secretion part over here. So what is secretion actually? We saw that absorption was absorbing material from the fluid in the tubule lumen. Secretion is exact opposite of it. That is materials, ions, solutes, which are there in blood capillaries and which is there in the cells of the renal tubule. They are secreted back into the fluid in the tubule lumen. So substances like hydrogen ions, potassium ions, ammonium ions, creatinine, which is a byproduct of creatine in the muscle, certain drugs like penicillin, they are secreted back into the tubule lumen for their excretion. Other substances, and the importance of this tubular secretion is like the secretion of hydrogen ion, it helps in controlling blood pH, and the secretion of other substances. That is how they are eliminated out of the body. So secretion is an important process. So over here we'll be discussing about the secretion of hydrogen ion. So what happens is CO2 is there in the fluid in the tubule lumen. CO2 is also there in the blood, in the blood capillaries. So this CO2, it diffuses inside the renal cells. This is the renal cell. Some of the CO2 is already present in the cell because of the metabolic reactions occurring in the inside the cell. So this CO2 reacts with water, and this reaction is catalyzed by an enzyme called carbonic anhydrase. This we may have studied in respiration chapter. So carbonic anhydrase, this leads to the formation of carbonic acid, that is H2CO3. This carbonic acid further dissociates into hydrogen ion and HCO3 ion, which goes out into the interstitial fluid, and HCO3 ions are preserved because this is an important This acts an important buffer system to maintain the blood pH. So what happens is this is an example of an antiporter, as we were discussing in previous slide. It was symporter. This is an antiporter. Antiporter, how it works is it transport one molecule from higher concentration to lower concentration in one direction, and other solute from higher concentration to lower concentration exact in opposite direction. So the sodium ions, as we know, because of sodium potassium pump, the concentration is lower inside the cell. So it is taken by this antiporter from the fluid in the tubule lumen and transported back into the cell. And hydrogen ion, as it is being created or produced by this H2CO3 dissociation, the hydrogen ion is in higher concentration inside the cell, so it is transported back into the tubule lumen. This is how hydrogen ions are secreted back into the tubule lumen. Other substances are also secreted by but different mechanisms. Different transport proteins are also there. So this was one of the examples of tubular secretion. So the next part is absorption in tubule in loop of Henle. This part is loop of Henle. This is just a second. So after PCT, the loop of Henle starts. This is the descending limb of loop of Henle, and this is ascending limb of loop of Henle. What happens is in descending limb of loop of Henle, this is impermeable to solute reabsorption. So no solute is reabsorbed in descending limb of loop of Henle. It is only permeable to water, and the type of water reabsorption which takes place is obligatory water reabsorption. We have studied this in previous slide. Now comes ascending limb of loop of Henle. In case of ascending limb of loop of Henle, it is exclusive to solute reabsorption. It is not permeable, that is, or we can say it is impermeable to water. No water is reabsorbed in ascending limb of loop of Henle. So, in order to understand how different solutes are reabsorbed in ascending limb of loop of Henle, this is the diagram. We have taken an example of sodium, potassium, and chloride symporter. This is a symporter protein over here. It is transporting one sodium ion, two chloride ion, and one potassium ion, transporting inside the cell. Because of the continuous function of sodium potassium pump, the concentration of sodium inside the cell is always low. So it is transporting the sodium from higher concentration to lower concentration inside the cell. This sodium is further transported out into the tubule lumen. Sorry, out in the interstitial space, in the interstitial fluid, through which the blood absorbs sodium into the blood capillary. For chloride and potassium ion, what happens is, for chloride ion, the leaky channels are there, but they are exclusively present on the basolateral side. This one. They are exclusively present on the basolateral surface. And for potassium ion, the leaky channels are exclusively present on the apical membrane. This one. The movement through leaky channels takes place from always from higher concentration to lower concentration. So the ion moves from higher concentration to lower concentration through leaky channels. The chloride ion, as it is transported inside the cell because of this symporter ion, it leaks out of the cell because inside the cell it is in higher concentration because it is transported inside the cell. And in the interstitial fluid, the concentration of chloride ion is low. So from cell and from inside the cell, it is transported or it leaks out of the cell through the leaky channel into the interstitial fluid. Ultimately, it is taken up by the blood in the blood capillaries. But with potassium ion, what happens is as the leaky channels are exclusively present on the apical membrane, it leaks out through this leaky channels back into the fluid in the tubule lumen. So they are coming back into the tubule lumen. They are not being reabsorbed. Ultimately, only one sodium ion and two chloride ion gets reabsorbed inside the blood. So one positive charge, two negative charge. Because of this, there is a net negative charge build up inside the blood. This net negative charge promotes the reabsorption of other positive charge ions like sodium, potassium, chloride, magnesium. These are positive charge ions, but their reabsorption is through paracellular mechanism. That is, they leaks out of these tight junctions between the cells and ultimately gets absorbed by the blood in the capillaries. So this is an example of reabsorption or absorption which is taking place in the in the ascending limb of loop of Henle, and no water gets reabsorbed in ascending limb of loop of Henle. Next is next is absorption in distal convoluted tubule, and collecting duct. This portion after the ascending limb of loop of Henle, this is distal convoluted tubule and this is collecting duct. In distal convoluted tubule, what happens is sodium ions are ultimately reabsorbed and potassium ions are excreted back into the tubule lumen. And how this happens is we have seen this in multiple slides before that the sodium potassium pump is present on the basolateral surface. We have cleared this concept that they're exclusively present on the basolateral surface and they're continuously pumping sodium out of the cell, potassium in, and pumping potassium inside the cell. So in the distal convoluted tubule and collecting duct, leaky channels, these are the leaky channels, are present for sodium ion exclusively on the apical surface, this over here. And for potassium ion, the leaky channels are present on both apical surface and basolateral surface. This. So because of this sodium potassium pump, whatever sodium is there inside the cell, it is going out. So there is a net lower concentration, constant lower concentration of sodium ion inside the cell. And because of this, the sodium which is there in the tubule lumen, it moves from higher concentration to lower concentration inside the cell. Continuously, this is occurring and further transported outside of the cell into the interstitial fluid and ultimately getting reabsorbed into the blood in the capillaries. In case of potassium ion, which is getting transported inside the cell, most of the sodium ion it leaks out through the leaky channel which is present on the apical membrane, this one. And very small amount of potassium ion goes out through the leaky channel which is there on the basolateral surface back into the tubule, back into the interstitial fluid. So maximum of potassium ion are going back or secreted back into the tubule lumen. So this is how the potassium ion, we may have studied this in the books, that the potassium ions are excreted out by the distal convoluted tubule. This is how it happens. The potassium ions are excreted out and the sodium ions are reabsorbed in the distal convoluted tubule. And ultimately, the urine which is formed through collecting duct, it is drained into a common papillary duct and which drains out the urine into the renal pelvis and goes out of the kidney through ureters. And ultimately, it is excreted out of the body through urethra. Thank you. Thank you so much.